So bioinformatics is kind of like a unique field where you can apply engineering principles to study basic building blocks of life, which I think is super cool. And then also you get to tie in all these different subjects like calculus, statistics, biology, physics, chemistry, everything is kind of mentioned. So I actually work mainly on phylogenetics, which I guess have people learned about evolution yet? Okay, you never know. So yeah, so I study evolution. So basically, how do we mathematically model it? How can we infer evolution from sequences? Stuff like that. Yeah. So real quickly, I'm going to talk about how can we apply proteins to phylogenetics. So phylogenetics needs some type of history of sequence mutation to be able to infer what is the evolutionary history. And it turns out that proteins are super good for this because if you kind of think about it, there's some sequence way back when, and then over time it evolves to present day. You can either have too little mutations, so if you imagine some sequence didn't change at all, if I have a bunch of sequences from present day, you can't really figure out what was the history. Whereas if it changes too much, you also can't figure it out because they all become constrained. So proteins are this really nice field where it's this like very nice middle ground of not too much mutation, but not too little, such that we can still study. But mainly today we're going to talk about protein structure, which is I guess what you guys are doing in your project, which is albeit a lot more cool, so that'll be a lot more fun. But first, some basic biology just to get everyone on the same page. How many people have heard of the central dogma? Whether you like fully understand it or maybe no one's heard of the central dogma. Okay, one person, nice. Woo! Okay, so this is a good review then. Basically, central dogma is kind of the mechanism of information propagation in the cell. So everyone knows what DNA is? Yeah? Okay, cool. So DNA is kind of like the blueprint of everything in life. So all organisms in current day have all of their genomes, which basically have all the instructions for every protein, every gene, all in one. But the DNA itself isn't functional. It just kind of is there as an instruction manual. So DNA is then transcribed to RNA, which is kind of the more functional but still more informational unit. And then RNA is translated into protein. And protein is really the functional kind of robot of the cell. It's the one that actually gets stuff done. So you have this kind of forward propagation of information from DNA to protein. And that's what we're really interested in. So kind of one question that people have is, if I have some protein that I'm interested in, how do I figure out what is its sequence? Because a protein sequence tells you a lot about the information of what it could do, where it came from, what species it is from, stuff like that. So there's a technology called mass spectrometry that basically you take your protein, that's just this kind of like three-dimensional funky shape, and you just chop it up as much as possible, and you can basically measure every single mass and try to reconstruct it. So basically the biological question here is, given a protein and given this weird shape of numbers and stuff, how can I reconstruct what was the actual sequence, which seems super abstract. And this is kind of the first motivation of why we need engineering in biology. But yes, the first question that I have is, given a protein, how can I reconstruct the sequence? Or, yeah, given a protein to reconstruct the sequence. The second question is basically proteins, they're these kind of blobby shaped things, and the shape that they make is kind of what defines the structure, the function that they have. So one example I love to show is this protein called cystic fibrosis transmembrane conductance regulator, CFTR. How many people know what cystic fibrosis is? Okay, so what is cystic fibrosis? Exactly, so it's a genetic disease that results in a buildup of mucus that makes it super hard to breathe. And it's actually entirely caused by mutations in this very single protein. Basically, this protein is what's called an ion channel. It's this kind of tunnel in your cell that helps things go from inside of the cell to outside the cell. And what I really like about this protein is the left is kind of this theoretical diagram that people try to help explain it, but the right is actually what it physically looks like. And if you kind of look at the right side, you see, especially in the left-hand one, these kind of tubes that form just three-dimensionally. So it's really interesting that just looking at the shape of it can tell you, oh, probably something has to funnel through this, if that makes sense. So yeah, protein structure dictates its function. So then, obviously, the question that we want to ask is, how do I figure out the structure of a protein? So there's a technique called X-ray crystallography that basically, if you have a protein just fixed somewhere, you can shoot a bunch of X-rays at it, and then you can kind of think of it as like a shotgunning effect, where like if you shoot a shotgun, the shell's going to fly all over the place. If you shoot X-ray proteins, basically the X-ray kind of scatters all over the place, and then depending on the protein, each shape of the protein dictates the unique pattern of these little dots that you get when it projects out. So then it turns into this question of, given these little dots, can I figure out what was the original structure of that protein? That's going to be the second question that we're going to investigate. Given this kind of abstract information, can I figure out what is the three-dimensional structure? Any questions? Okay. So the third question is basically, if I have some unknown protein sequence, can I also try to figure out its function? So a sequence of a protein is a lot of times all you have to work with. You don't always have that three-dimensional shape, and it's a lot harder to build. So if I had this massive repository of a bunch of sequences, it would be useful if I could just figure out what is the function of every one of these proteins. So that's another biological question that we're going to tackle in a little bit. And then another question is also, instead of the sequence, if I'm given the structure of the protein, just like that kind of tunnel shape thing, can I figure out just from the shape of the protein, what might its function be? And then the last question that I want to dive into deals with basically how proteins work themselves. So have people learned about proteins at all? No, kind of. Yeah, so proteins basically, like I mentioned before, they're the functional unit of the cell. So proteins, each protein has a specific function that it has to do. And then the cell wouldn't be able to survive if a specific protein didn't do that function. But a lot of times when you read a biology textbook, they kind of say, this protein goes to this part of the cell and does this. And it makes it sound as if proteins are kind of robots that know what they're supposed to do, which is actually not at all the case. Proteins are actually what are called stochastic. They're these random things that basically float around in the cell, just kind of shaking around. And then coincidentally, if they by chance hit what they're supposed to be hitting, then some reaction occurs. But they don't actually target it, go towards anything. They just kind of float around randomly. So another question is basically, if these proteins are just floating around randomly, and everything in the cell that's happening is so random, can we figure out if I design a drug, can I have it randomly hit a protein that I want and also not randomly hit other proteins? So if you guys have ever seen those advertisements for medicine, you know that the actual commercial has 10 seconds of actual information, and then it's 30 seconds of someone saying a bunch of side effects, right? So side effects in a medicine is specifically this concept of, if I give myself some drug, this drug is supposed to be targeting one protein, and by targeting that protein, it kind of fixes whatever disease I have. But if it accidentally hits something else in the cell, it causes some downstream ripple effect that I might not want, and that is the basis of side effects. So another more medical question is, if I'm designing some therapeutic, and I know what protein I'm interested in, maybe for some reason this is related to disease, and I know what proteins exist that I'm not interested in, can I design a drug that specifically targets my protein and not any of the others? So basically, can I minimize any side effects? Does that kind of make sense? Yeah. So if you think about it, in this case, this little arrow is kind of the drug that I'm designing. If I can somehow simulate, does it fit well with this one, but hopefully figure out it also fits well with this other one, maybe I can do some more targeted design. So the next biological question is basically, yeah, given a bunch of structures of proteins that I know, and given that I know which ones are good and which ones are bad, can I somehow figure out if some drug I'm designing hits only the good ones and not the bad ones? Okay, so any questions about the biology? I kind of breezed through that just because I didn't want to go super deep, but yeah, any questions? Yeah. Can you repeat that? Sorry. Yeah, okay, so the question is basically, do all drugs have these off-target effects? Are none of them perfect, and do they all accidentally hit some off-target protein? And the answer is usually, yeah. Usually, every drug that I've seen, that's why you get these commercials that have 20 minutes of just side effect after side effect after side effect. Because as far as I'm aware, there is no drug that perfectly hits a specific protein. You can have stuff like insulin, but insulin is a protein that does very targeted. But yeah, in general, they're pretty bad. Yeah, any other questions about biology? Okay, so now tying back to the engineering. So in the previous part, I mentioned all of these biological questions that we want to answer, but it turns out that you can't answer them without actually applying engineering principles. So the first question that I asked is, basically, if I have some protein and I break it up every single possible way, and I basically measure the mass of all the fragments that I generate when I break it up, can I somehow reconstruct the sequence of that protein? And it turns out that you can turn this into basically an electrical engineering graph problem. So you can design these masses as networks, and then figure out, is there a path in this network that I construct that explains these peaks? That path defines the sequence. So I'm not going to go into what actually is happening here, but basically you can transform this very theoretical biological problem into a very specific computer science and engineering problem. And then once you solve that problem, you've also solved the biological problem. And the second thing, basically, if I have an unknown protein and I shoot this X-ray at it and I get the scattered shape, can I figure out the original structure? Basically, again, this is an engineering question. The question is, from these little dots, can I somehow figure out what had to hit them to be able to get this weird shape? And it turns out that this is also something that just turns into a physics problem. So what people did was they noticed, if I shoot this X-ray at this object, basically the angle at which it bounces off is defined by every single X, Y, Z position of the protein. So I can basically take my dots and then kind of reverse engineer what has to be every single X, Y, Z for every single dot to make it, if that kind of makes sense. So again, I'm not going to go into super details about how they do that, but basically you just do very, very complicated physics, and you can actually reverse engineer and structure the protein just from those little dots. Another question that I didn't really mention is, I mentioned we wanted to reconstruct the structure, and I mentioned that we had X-ray crystallography to do this. It turns out that even from just the sequence, you can also predict the three-dimensional structure. I'm not really going to go super into this because this is a little complicated, but basically, yeah, you can try to also, instead of figuring out the exact structure, you can try to predict it just from the sequences. The third question that I asked was, given an unknown protein sequence, can we figure out its function? Basically, given the sequence, can I try to figure out what does it do? So the solution to this, again, it's a computer science question. Basically, if you have a bunch of sequences from organisms that you already know what they do, you can try to see this unknown sequence, which sequences does it look the most like? If you have some family of sequences that all perform some specific function, and you see that your unknown protein happens to fit really nicely with them, that kind of implies that your protein is probably in the same family. So you can try to figure out what might be the function of your protein just by seeing what it looks like, if that kind of makes sense. So it's kind of guilty by association type thing. You can figure out what it does based on what it looks like. And the fourth question I asked was, given an unknown protein structure, can we also figure out its function? So like the tunnel shape thing I mentioned, the shape of that tunnel kind of tells you it's probably something that's going through it. It turns out we can also do the same thing where we look at other protein structures that we've already figured out, and we try to see three-dimensionally what does our protein look like the most. Whatever that protein does, our protein also probably does. So you can kind of infer from already known information, what does this new protein actually do? And then that last question that I was talking about with respect to drug design, basically if I have a bunch of protein structures, and I have a structure of some therapeutic that I want to create, and I want to see what therapeutic can only hit the genes that or the proteins I'm interested in, and not hit anything else. It turns out we can also figure that out. So like I mentioned, proteins, they just kind of like shake around in three-dimensional space. So in here we have a protein, which is the kind of ribbony shape thing. And then we have a potential drug, which is that kind of hexagon floating shape thing. And using what we call molecular dynamic simulation, so basically just simulating how does this protein move in 3 dimensions, we can figure out how well does this drug attach to this given protein. So if I was trying to design a drug, what do you guys think I should do? Does anyone have any ideas? How would I design a drug that works the best? Any ideas? Hey, what's up? That's here. Yeah, so he made a good question. So the answer was basically, we can try to design a drug that hopefully we design to fit nicely in that empty space in the protein. That's exactly right. You can try to design a drug that basically, in the simulation, when it hits your protein of interest, hopefully it kind of stabilizes and fit like a glove in that little hole. So that's basically what research is when you're designing a new protein, or a new therapy. And then also you can test, is this drug that I made good by testing against all the bad proteins and making sure for every single bad protein, it should not fit very well. So yeah, that was all the kind of technical details I wanted to give. I went, I guess, a little fast. But basically, my motivation to you all is you should learn bioinformatics if you can. Just basically, it's fun to learn a lot of different things. And in bioinformatics, you get to pretty uniquely learn about chemistry, physics, calculus, statistics, computer science, so many different topics. Also, if you are purely interested in biology, I still think it's useful to learn bioinformatics just because all of biology is moving towards these super big DNA sequencing data sets. And it's kind of hard to only go in with just biology. You kind of have to know a little bit about how do I do basic data analysis to be able to function. And then also, it gives you a lot of freedom with respect to what you want to do with it. So right now, I'm doing mathematical modeling, but I've also in the past done more like medical, directly related to the drugs type research. I've also done more like working with worms and stuff like that. So basically, with the bioinformatics background, you have the skills to basically try to do whatever type of research you might be potentially interested in. And then why study proteins specifically? You know, proteins, they're the functional unit of life. If you understand how a protein works, you for the most part can understand how exactly a cell works. You can understand the mechanisms of whatever biological pathways you're interested in, and you can understand why exactly does a disease happen. And if you understand why the disease happens, you can figure out how can I intervene and prevent it from happening. And then you can also design more effective drugs, and you can learn about how species evolve over time and how like life evolves, I guess. Yeah. So that was my quick blurb on proteins, but I want to kind of leave the floor for any questions that people might have about pretty much anything. Any questions?
Talking about diseases and drugs that are designed for diseases. So you get to choose a protein. Then you're going to have to design a model for that protein. So what he was talking about are ways that bioinformatics is looking at the information behind the biology and figuring out how can you design a drug to help for a disease. Or how does this part of our body work? So you get to pick something that you're interested in, and then you're going to design a model. So those of you that are engineering, you're working with these amazing 3D printers. So 3D printing is a really new technology. In biology, they're using it to even build body parts. So literally laying tissues together to build a new ear for someone. Or in engineering, you're using plastic pieces and you're layering the plastic to build a piece. And that's what you guys are going to do. You're going to use the 3D printers to design your protein. Okay? So where we're going to move from this is into understanding how these proteins work and then building those proteins. Okay? So I'm going to ask you to stay where you're at for now. You need to check in if you haven't done, if your 7th grade teacher is in this room, you need to check in for attendance with your teacher unless, Ms. Rasmussen, did you already get everybody? Okay, Mr. Cannon? Mr. Cannon. Did you get everybody for your 7th period? Did they check in with you for attendance? Okay. So my guys.
Hey, why not, guys? 4 minutes here.
Ladies and gentlemen, I'm going to ask you one more time.